My name is Regina O'Kelly, and I'm a PhD candidate at Oregon State University in the Soil Science program. My advisor, Dr. David Myrold, and I are leading the Willamette Valley study in the NRCS Dynamic Soil Properties for Soil Health Initiative. Like the other cooperators, we are measuring a wide range of soil properties in the study, but today I'd like to focus on just a few involved in the carbon cycle. Something I really enjoy about soil health research is that it gives us the opportunity to look at soil properties as indicators of ecosystem functionality and think about how they can complement each other. The dynamic soil properties concerning carbon are a great example of how soil health research has developed over time and helped us to gain insight into microbial processes in the soil. However, there's still some uncertainty about how some of these properties fit together. So I would like to tell you a bit about one of the properties we measured, permanganate oxidizable carbon, and share some of our results so far. So soil organic carbon has been considered important in the soil for a long time. It functions as food for microbes. It supports important soil functions for plants like nutrient storage and aggregation. Organic carbon in the soil is a cornerstone of the science of soil health, but measuring it in a way that's helpful for management is actually pretty complicated. Soil organic carbon as a whole can be fairly slow to respond to ecosystem disturbances and management decisions, which is a drawback as a soil health indicator. Useful soil health properties should function as early indicators so that land managers can adjust their strategies quickly. We also need soil health indicators to be relatively quick to run and inexpensive to make soil health monitoring practical. Over the last few decades, permanganate oxidizable carbon, or POXC, has become a popular soil health indicator because it checks a lot of these boxes. The method has been fine-tuned over time to be less hazardous, faster, and more consistent, but I'm just going to give a super quick summary of how it's done. The basic process is that 2.5 grams of air-dried soil is shaken with a dilute solution of potassium permanganate and allowed to settle for 10 minutes. The permanganate oxidizes carbon in the soil, and the solution becomes lighter as it reacts with the carbon. The more carbon there is in the soil, the lighter the solution will be at the end of the reaction, and you can estimate how much carbon there is by the end color of the solution. This is typically done with samples taken back to the lab, but there is also a field version available to estimate carbon without sending samples away. You may also hear POXC called active carbon. That's because, in addition to being sensitive to management, many studies have reported that POXC is actually a specific fraction of soil that is more easily accessed by microbes. So it's cycled faster or more actively than the rest of the soil organic carbon pool. So if that's the case, we would expect to see that POXC is associated with properties related to soil microbes, like the respiration rate or other microbial activity measurements. There are a number of studies that have reported that POXC is correlated with some of these properties, but it's not always the case, and the strength of those relationships vary across studies. Given this history with POXC, I wanted to look at how strong these relationships were in our Willamette Valley sites and evaluate two of these major arguments for using POXC. I looked at how much POXC varied between management systems and compared POXC to the respiration rate, which is one way we're measuring microbial activity in this study. So before getting into results, I want to tell you briefly about the study design. So we are measuring soil health indicators on two extensive soil series in the Willamette Valley. The Woodburn series, which is a mollisol found on terraces lower down in the Jory, lower down in the valley. And we also have soils from the Jory series, the official state soil of Oregon, which is an altisol found farther up in the foothills of the valley. I want to emphasize here that because these two series are so different from one another, I'm comparing management systems within each soil to each other, but not between the two soils. The two soils are going to have different properties that are more or less inherent to them. So it's important to look at how properties within them vary to avoid mixing up the effects of management with the effect of soil formation factors. On both soils, we have perennial crops with a range of management conditions. On Woodburn, we took samples from mature hazelnut orchards and perennial grass seed fields with two different levels of tillage. These are mostly perennial rye, with a couple sites with oatgrass and tall fescue as well. Jory is relatively commonly found in forests, so as our lowest intensity management, we sampled from second growth timber stands. The Willamette Valley also has a very active wine industry and Christmas tree industry. So our Jory sites also include vineyards and Christmas tree farms. As you can see, the Willamette Valley is pretty diverse in terms of the different crops growing here, and each of these management systems has a range of different characteristics that can influence the soil. So because these management systems are so different, I was a bit surprised to see that POXC did not differ between these different management systems. Here we have POXC along the soil profile in the first 100 centimeters, with the mid-horizon depth on the vertical axis and POXC along the horizontal axis. So there's quite a bit of variability within depths in many cases, but overall, depth is a much stronger influence on POXC than management is. The total soil organic carbon, however, does differ by management systems. So when I normalize POXC by the total soil organic carbon, meaning expressing POXC as a percentage of soil organic carbon, there is a difference between management systems. These box plots are showing that percentage in the 0 to 5 and 5 to 10 centimeter horizons. And when we look at the data this way, in vineyards and Christmas tree farms, a higher percentage of the soil organic carbon is POXC than in forests. Within the Woodburn sites, the differences are more subtle or non-existent for the 5 to 10 centimeter horizon, but there is more sensitivity to management than when you're looking at POXC just on its own. So to interrogate the claim a little bit that POXC represents a relatively active fraction of organic carbon, we can see how it relates to the pool it's part of, soil organic carbon, and whether it functions as a good predictor for the microbial activity by plotting these variables against one another. I was able to confirm that POXC is closely correlated to organic carbon in both the Jory and the Woodburn series. If POXC is some fraction of organic carbon, we would expect to see that the more organic carbon there is, the more POXC is present. Depth is also a control here for both variables, which is pretty typical as far as previous work goes. Soil organic carbon doesn't explain all of the variability in POXC, but overall POXC is so far behaving as we would hope, a practical index of soil organic carbon across management systems in both soils. Now let's see how well it indicates microbial activity by comparing it to another common dynamic soil property, mineralized C. The vertical axis in the next few graphs is the respiration rate during a 4-day lab incubation, the mineralized carbon. And I'll show you two different ways to express POXC on the horizontal axis. In the first set, POXC is simply expressed as a concentration, milligrams of carbon per kilogram of dry soil. In this case, POXC is still fairly closely related to the mineralized carbon. The respiration rate increases with POXC. This relationship is a bit tighter in the Jory sites than in the Woodburn sites, and including depth in the model improves this relationship as well, along with management. This offers good support for POXC as a relatively rapid predictor of microbial activity if it's not practical to measure mineralization directly. We can play the same game with organic carbon, plotting the percent organic carbon against mineralization, and we see the same trend. I already told you that POXC increases with organic carbon, so it isn't a huge surprise that respiration increases with both of these measurements of carbon. However, organic carbon is actually slightly more correlated to mineralization than POXC if you compare the R-squared values on the relationship. This is inconsistent with the idea that POXC represents a particularly available fraction of carbon. If that were the case, we would expect to see a better relationship between POXC and respiration. I would even argue that there should be a relationship between POXC normalized by organic carbon and the respiration rate. If there's a greater fraction of the organic carbon that's this more active available fraction, then that percentage should be related to the respiration rate, I would think. So that's the comparison I've plotted here. By normalized in this case, I mean that I've expressed POXC as a percent of the organic carbon. So the horizontal axis is showing what percent of the organic carbon is permanganate oxidizable carbon. And there's basically no relationship between organic carbon normalized POXC and respiration in either soil. This is true both considering all of the samples together and after accounting for management. You can see that by the color, the depth is a much stronger control in respiration than POXC is, or POXC normalized by organic carbon anyway. So while POXC does correlate with microbial activity and total soil organic carbon in these series, there isn't very strong evidence that it represents a particularly available fraction of carbon for microbial activity. So what's going on here? Well, it's a bit too soon to draw conclusions, but there is some previous research that might offer some insight. So some researchers have compared POXC to other carbon fractionation techniques and found that it does correlate well with more processed fractions of carbon, as well as practices that increase carbon sequestration rather than mineralization. This 2016 paper I've referenced here argues that if that's the case, we wouldn't actually expect POXC and carbon mineralization to be perfectly correlated. They would function more as complementary measurements showing the balancing forces of mineralization versus stabilization processes. However, I have not measured those other carbon fractions on these soils, so I need to express really strong caution in associating that process with the results here. It isn't clear whether that's the mechanism behind the results in the Willamette Valley soils, but it's perhaps a good place to look into further. So despite this and the other surprising result that POXC was not very sensitive to management, some of my results do support using POXC as a soil health indicator. POXC did have a strong relationship with both respiration and soil organic carbon. So even though it isn't clear whether POXC represents a particular type of carbon, it does seem to function well as a faster and fairly inexpensive alternative to these properties in the soil, although both soil organic carbon and respiration were less variable among sites. I see balancing these practical factors against other study considerations as a really important area of research in soil health, and I'm hopeful that we'll get a little closer to finding that balance throughout this project. With that, I would like to acknowledge the other folks involved in the Willamette Valley study, my advisor, Dr. David Myrold, who is the PI for this study, and research assistant Cedric and Lucas, who have helped collect a lot of these data. This research would also not be possible without funding support and leadership from NRCS. And finally, thank you so much for tuning in.